Ima Elisha Di Arif, a second-year student from MSc Desidio, currently taking up Bachelor of Science in Marine Science. Today, I will be discussing about protein synthesis as part of our requirements in biochemistry under Dr. Efren Tangon. By the end of this video, you will understand how our cells use DNA instructions to build proteins that keep us alive and functioning. Let's get started. To begin, let's quickly talk about DNA and proteins. DNA is like a giant instruction manual inside almost every cell in our body, and proteins are molecules that carry out instructions written in our DNA. They're involved in everything from building tissues to making enzymes and hormones. But how does DNA actually turn into a protein? DNA turns into a protein in a process what we call protein synthesis. Protein synthesis is a biological process in which cells build protein. It is a complex multi-stage process that can be broken down into two major stages, transcription and translation. Let's explore each step starting with transcription. Transcription is the first stage of protein synthesis, where the cell copies genetic information from DNA into messenger RNA or mRNA. The template for messenger RNA is a genetic sequence along a section of DNA strand. Each strand of DNA contains nucleotides with complementary base. Adenine pairs with thymine and cytosine with guanine. This process occurs in the nucleus of the eukaryotic cells, where it involves several steps. The first step is initiation. During initiation, the double-stranded DNA unwinds with the help of the enzyme called RNA polymerase. This enzyme binds to a region of DNA called the promoter, marking where the transcription should start. The second step is elongation. During elongation, RNA polymerase moves along the DNA, reading its sequence and building a complementary strand of mRNA. In RNA, thymine is replaced by uracil, so adenine pairs with uracil instead. Then we go now to the third step, which is termination. The RNA polymerase continues until it reaches a termination sequence, signaling the end of the gene. The completed pre-mRNA strand detaches containing both exons, which are coding sections, and introns, which are non-coding sections. And the fourth step is RNA processing. Once the mRNA is built, certain enzymes remove introns, or the sections that will not be used to build the protein. Enzymes splice the remaining ends or exons together. The functional mRNA leaves the nucleus and heads to the cytoplasm for the next phase, which is translation. Translation is the process where the genetic code carried by mRNA is converted into a specific sequence of amino acid, forming a protein. This occurs in the cytoplasm at the ribosome, which can be free-floating or attached to the endoplasmic reticulum. Translation consists of initiation, elongation, and termination. Initiation. First, the ribosome assembles around the mRNA. A transfer RNA or tRNA carrying methionine binds to the start codon AUG. The large ribosomal subunit then joins the small subunit, and we're ready for elongation. Elongation. During elongation, the ribosome moves along the mRNA. The tRNA brings in the correct amino acid, which are linked by peptide bonds to form the growing protein chain. This continues with the ribosome shifting along the mRNA and adding amino acid in sequence. Termination. Finally, elongation continues until the ribosome reaches a stop codon on the mRNA. A release factor binds to the ribosome, freeing the newly synthesized protein, and the ribosome disassembles. This mRNA can now be reused for additional round of translation if needed. Protein synthesis also undergoes post-translation modifications. After synthesis, the protein often undergoes additional steps to become fully functional. It might fold into its specific three-dimensional shape, sometimes with the assistance of chaperone proteins. So to recap, Protein synthesis starts with the DNA in the nucleus, where it's transcribed into messenger RNA or mRNA. The mRNA then travels to the ribosome in the cytoplasm, where it translated into chain of amino acids. Finally, the chain folds into a protein, ready to perform its specific function